In the complex world of cellular biology, a silent battle rages within our membranes. It is called ferroptosis, a form of programmed cell death triggered by the oxidation of lipids. This process is a double-edged sword. While its overactivation can lead to neurodegenerative diseases, its suppression is a primary weapon. To survive, tumors rely on two guardian proteins, GPX4 and FSP1. These proteins act as shields, halting the chain reaction of lipid destruction. Recently, a breakthrough study from the University of Würzburg has revealed a surprising regulator, vitamin B2, also known as riboflavin. Scientists discovered that FAD, a metabolic product of this vitamin, is not just a helper. It is the very scaffold that maintains its three-dimensional shape. Without sufficient riboflavin, the FSP1 protein becomes unstable and simply degrades. This leaves the cancer cell completely exposed, rendering it highly susceptible to the very death it tried so hard to avoid. To prove this link, researchers used CRISPR gene screening on human fibrosarcoma cells. By knocking out the primary defense, GPX4, they created a cell model. They found that removing the enzymes responsible for processing vitamin B2 caused FSP1 levels to plummet. Proteomic analysis confirmed the truth. FSP1 is one of the most sensitive proteins. When the vitamin supply runs dry, the cell's final line of defense vanishes. This discovery led to an ingenious experiment using a natural antibiotic called riboflavin. Think of riboflavin as a molecular mimic. It looks almost identical to vitamin B2, allowing cancer cells to absorb and process it into a fake version of FAD. This fake molecule binds to the FSP1 protein, successfully stabilizing its structure. However, there is a catch. This decoy version of the protein is completely non-functional. It cannot accept electrons or stop lipid peroxidation. The cancer cell is tricked into keeping a broken shield. Even at remarkably low concentrations, this induced ferroptosis across melanoma, breast, and lung cancer lines. Simultaneously, researchers at UC Berkeley identified the cleanup crew, an enzyme called RNF8. This enzyme acts like a quality control inspector, specifically identifying and destroying FSP1 proteins. Together, these studies complete the mechanical chain, showing exactly how we can manipulate the cell's internal logic to trigger its own demise. These findings lay the foundation for new anti-cancer drugs targeting the riboflavin pathway. But there is a final, crucial realization. Most lab-grown cancer cells are currently raised in a honeypot, with vitamin B2 levels 50 times higher than those in the human body. To find the next generation of cures, we must move beyond these artificial environments. The path to defeating cancer may just be hidden in the vitamins we once took for granted.